Yes, madam. Good morning, madam. Myself, Dr. B. Govindarajulu, Associate Director of Research.
Good morning.
We are All India Coordinated Research Project on Citrus, Rayalaseema Zone, Tirupati, madam.
Okay.
Good morning, madam.
Good morning, madam.
Ruth, madam.
Dr. Ramaya, madam. Good morning, madam. Dr. Ramaya, madam, Principal Scientist and Head, madam, Citrus Real Estate in Petlur, madam.
Oh, okay. Nice meeting you.
Good morning, Govindrajulu, sir.
Madam,
Sir,
very good morning.
Please unmute yourself, sir. You are not audible.
Yes. It is audible now? It is audible?
Audible, sir.
Ruth, madam. Good
Sir,
morning.
sir, audible వినిపిస్తుంది, sir.
Okay, fine. Madhavi, madam, good morning. Madam, వినిపించినట్లేదు.
Madam వినిపించట్లేదు. ఎంతమంది జాయిన్ అయినారు?
Madam, good morning.
Good morning.
Dr. Chandrika Ramudugu.
Good morning, Aunty.
Good morning, good morning.
VC sir, good morning.
Good morning.
University officers. Madam, we will start in 5 minutes, madam.
No problem.
Sir, good morning, sir.
Good morning, madam.
Okay.
Sir, good morning, sir. Dr. Ramaya, sir.
Good morning, good morning.
Sir.
Morning, sir. Good morning, Chandrika madam. I am Dr. C. Madhumati. We met in that citrus National Citrus Workshop at Jalgaon, madam. Nice to meet you.
తీసేయలేదు. మీరు ఇటు జరగండి.
Good morning to all of you and a warm welcome to respected Honorable Vice Chancellor of Dr. YSR Horticultural University, Dr. K. Dhananjay Rao Garu, and welcome to today's guest speaker, Dr. Chandrika Ramadugu, project scientist from University of California, Riverside, USA. And thank you, madam, for accepting my invitation to conduct the 27th Global Connect series. It is my privilege as convener to welcome all of you to this Global Connect series. The topic is citrus honglongbing, use of breeding genetics and genomics to develop novel varieties with disease resistance in collaboration with the University of California, Riverside, USA on 17th February 2026 at 10:30 a.m. under the chairmanship of Dr. K. Dhananjay Rao Garu, Honorable Vice Chancellor Dr. YSR HU Venkatramanagudam, West Godavari District, Andhra Pradesh, India. Thank you, sir, for giving permission to conduct the 27th Global Connect program under your dynamic and visionary leadership. It is my privilege to extend a warm and heartfelt welcome to our most respected Vice Chancellor, sir, and esteemed university officers, Dr. M. Madhavi Madam Garu, and Registrar of Dr. YSR HU, and Director of Research, Dr. Ramanandam Garu, Dr. Prasanna Kumar Garu, Dean of Horticulture, and Dr. Surya Kumari Garu, Dean of PG Studies and Controller, and Dr. Vijay Padma Garu, Controller of Examinations and Director of Planning and Monitoring Cell, and Dr. C. Madhumati Madam Garu, University Librarian and Dean of Student Affairs, and Engineer Ramana Garu, Estate Officer of Dr. YSR HU, and all the Associate Director of Research ADRs of LAM and Tirupati, and all Associate Deans of Horticultural Colleges and affiliated colleges, and Principals of Horticultural Polytechnic Colleges, and all the heads of Horticultural Research Stations and Krishi Vijnana Kendrams, and the rapporteurs of this program, and other participants, and my dear students of Dr. YSR HU. Welcome to this guest lecture, Citrus honglongbing disease or citrus greening, caused by Candidatus Liberobacter asiaticus, which is a dangerous and destructive disease and caused economic damage to the farmers in India and Andhra Pradesh and many districts. And before inviting our Honorable Vice Chancellor, sir, just I will give brief introduction about guest speaker. And Chandrika Ramadugu is a project scientist at the Department of Botany and Life Sciences, University of California, Riverside, California, in the United States of America. And Madam obtained her MSc degree from Mysore University, specializing in botany and plant pathology, and she obtained her PhD degree in microbiology and cell biology from Indian Institute of Science, Bangalore, India, and as a postdoctoral fellow in various laboratories in the United States. Chandrika Madam worked in animal viruses like measles and plant viruses like tobacco mosaic virus, and she was a chief scientist in a plant genetics company, Integrated Plant Genetics, developing plant transformation techniques for citrus, rice, and geranium crops. And Dr. Chandrika Madam worked exclusively on citrus for the past 20 years at the University of California, Riverside, USA, and she is the lead principal investigator on many multi-million-dollar projects aimed at developing sustainable solutions for the citrus honglongbing issue. Her scientific team consists of researchers from various organizations in the United States with expertise in several related fields of study. The research involved the development of full genome sequences of disease-resistant and susceptible genotypes, and identification of candidates resistant associated genes, genotyping, and developing methods to identify resistant plants, design of psyllid traps also for insect control of honglongbing vector, etc. Recently, the Indian Society of Citriculture has bestowed honorary fellowship in recognition of her contributions in the advancement of citriculture. This is a brief biodata of Chandrika madam. Now I am inviting our Honorable Vice Chancellor sir to give this his presidential address on this 27th Global Connect series. Please, sir.
Thank you, madam. Director of Industrial and International Program, Dr. YSR Horticulture University, Dr. C.H. Ruth Garu. Thank you. Madam, today guest speaker, Dr. Chandrika Ramadugu, Project Scientist, Department of Botany and Plant Sciences, University of California, Riverside, California, USA. Welcome, madam. Welcome to Dr. YSR Horticulture University. And very having pleasure to invite you and thank you for accepting our invitation from Dr. YSR Horticulture University, because this is the 27th Global Connect series we are conducting today. And already we have conducted 26 programs earlier, arranging international scientists, eminent international scientists, to update the knowledge in horticulture and agriculture. So thank you, madam. And moreover, it is a midnight for you, I think. You are losing sleep and want to give the lecture on the citrus, which is a very important crop. And all our esteemed university officers, associate directors of research, and associate deans, and all heads of research stations, principals of polytechnic colleges, and all heads of research stations, heads of KVKs, and associate deans of all colleges of Dr. YSR Horticulture University. And it is a very prestigious program we are conducting from YSR Horticulture University since 3 years. And this is the 27th program. And you know that citrus is a very important crop. It is grown in India almost 1100,000 hectares, and with the production of 14.6 million metric tons, and grown in 16 states of India. And in Andhra Pradesh is one of the important crops, sweet orange and lime. And in Andhra Pradesh, the production is 2.26 million tons of the data of 23-24. So this is one of the important crop, especially sweet orange. Mostly 90% of the crop by the Satgudi variety of sweet orange. And this is one of the important problem, that is greening, citrus greening. That citrus greening is a major disease also in Andhra Pradesh. And we have excellent research stations, citrus research station Tirupati and citrus research station Petlur. And in Mahanandi also we want to encourage the sweet orange and acid lime, and we want to produce the virus and disease-free plants, budlings. So we started screen houses at Tirupati and planning to have one more at Mahanandi. And you know that this is the citrus greening. General, it is caused by the bacteria, and that is transmitted by the bug, that is psyllid. So everybody thinks that greening caused by the virus, but it is bacteria, caused by the bacteria. The minimum symptoms like yellowing of veins, defoliation, dieback, stunted growth of fruits, yellowing of fruits, and finally death of the plant and fruit drop. This is the major disease in citrus. And in case of United States, that Florida citrus industry is getting affected with this disease. And some that varieties, we don't have resistant varieties so far. And with the biotechnology work, Madam almost done 20 years of research on citrus. And Madam also received the honorary fellowship in citriculture. And Madam is a botanist, pathologist, microbiologist, and 20 years of experience in citrus. And congratulations, Madam. And you have a lot of experience in citrus. And we are very fortunate enough to have a lecture from you. And that what is the molecular biology work is going, and excellent work is going as per the report I have received from you. And you definitely guide all our scientists, young scientists, and scientists, students. And because this is a program of entire state of Andhra Pradesh, constituting of almost 41, 42 institutes of Dr. YSR Horticulture University. And in this, all MSc and PhD students are also participating, all scientists are participating. So thank you very, very much once again. And please, you can proceed.
Thank you, sir, for your valuable message. And actually, citrus plant pathology was started in 1936 in Kadapa district and Tirupati district. Previously, many scientists worked on citrus diseases, madam. And now I invite Dr. Chandrika, madam, to deliver the topic on citrus honglongming disease. And please share her insights, madam. Madam, please start.
Okay. Can you see my slide?
Yes, madam.
Is it in the presentation mode?
No, madam, it is in slide mode.
Oh, presentation mode. Yeah, let me get some technical help. Yeah.
Okay. I'll try.
Yes, ma'am.
Yeah.
Now it's okay?
Yeah.
It is okay. Okay, madam.
Thank you.
Namaste and good morning to everybody. Thank you for giving me this opportunity to share our research with your esteemed group. So my name is Chandrika Ramadugu from University of California, Riverside. We have been working on citrus honglongming or citrus greening. And I'm going to describe some of the methods and concepts that we have to do this work. Can you see my second slide?
Yes, madam, go ahead.
That's good. So as you heard before, this is a bacterial disease of citrus. And the bacterium that is associated with it is called Candidatus Liberibacter asiaticus, which is an unculturable bacterium. It has a very small genome, so it can only survive in the plant or in the insect vector. And the insect vector is Diaphorina citri. You can see an image of the psyllid here, Diaphorina citri. When the plant is infected, you get a blotchy mottle, as is shown in the right panel here. That is, you have blotchy patches of yellow things, and also the symptoms vary depending on the genotype, depending on the variety you are looking at. The fruits are lopsided or of this shape, not uniform, and they develop a lot of off flavor because of HLB. Now in Florida, I lived in Florida for many years. Florida citrus is a major crop. And before HLB, what you were seeing was like this tree in the inset, you know. In October, the tree will be full of fruits and a lot of yield, and citrus is a major crop in Florida. But now, since 2005, the disease has come, and now in October, in the same region, this is what I saw a couple of years ago when I went to visit. And the tree has no fruits. Of course, in addition to Hang Lang Bing, they also have lots of hurricanes, which make the fruit fall. So anyway, the loss is tremendous. This is a map that shows that the acreage of citrus has gone down. The cost of managing citrus has gone up. And as of now, as of 2025, the yield that was obtained in 2025 was 5% of the yield that was there before the disease came in 2005. So it is a very serious problem. And as you know, the disease triangle, the host, the pathogen, and the conducive environment are three of the important things. In addition, we also have the psyllid factor. So you have to control many things before you can get some kind of disease mitigation. So we wanted to see if there are any citrus genotypes or citrus relatives that are resistant to this disease. So we have a citrus variety collection. I'm going to show you a picture of that after a couple of slides. So we took plants belonging to 22 genera, not just citrus, others that are close to citrus. And we took them to Florida from California and grew them in the field for like 6 years. This is a disease that you have to study for a long time. You cannot make conclusions in few months or a couple of years. So this experiment, field experiment, went on for 6 years. Then we determined that there were some genera that were totally immune to the disease, like the one here, Glycosmys pentaphylla. And there were some that were fairly resistant, like the Australian limes, Eremocitrus glauca, or the desert lime, Microcitrus australasica, or the finger lime, and Eagle marmelas, which is the Bilvapatra that we have in India, and the trifoliate hybrid and other citrus types. Basically, many of the citrus types are susceptible. What is shown here as S is susceptible. What is shown here as R is resistant. And the Australian limes are more resistant, are highly tolerant to the disease. One of the interesting things about citrus is that you can cross citrus different species. You can also cross closely related genera. And we found out that you can cross the Eremocitrus and Microcitrus types with citrus. So there was an opportunity for us to generate citrus that may have resistance. And we published this a few years ago. Now, why is breeding essential? Because no one likes to do tree breeding. It's long term and it is pretty challenging. The problem with citrus is that the crop has genetic vulnerability. That is, majority of the citrus cultivars are generally derived from a few varieties only, like your sweet orange or even grapefruit. All the grapefruit that we grow here basically comes from one variety, which is the Duncan grapefruit. Now, they all look different. They have different types of flesh and flavors and all that, but genotypically, they are very, very similar. So this kind of cultivation is called a monoculture. And when you have monoculture, there are advantages because maintenance is easier. You do the same thing for the whole area and all that. So, but when you have a pathogen, especially a new pathogen come in, that can be disastrous. So what can we do about this? You can do genetic enhancement. That is, you broaden the genetic base by incorporating new germplasm. So that's what we try to do. And in many other agricultural crops also, people use wild crop relatives to bring desirable traits, horticultural traits. So now when you cross citrus with a wild plant, naturally there will be many traits that you may not like. but you can introgress the trait that you like by crossing it back to citrus several times. So evaluation, I'm going to talk more about evaluation later. So that is the phenotypic assessment of the genetic potential of these novel hybrids. You have to do it in multiple environments because genotype by environment interactions are also known to be crucial. So citrus breeding provides opportunities and limitations. Now let's look at the limitations first. It has long generation times, 4 to 9 years. That's why it's been taking so long to do our work. Citrus also has many biological peculiarities, which makes things challenging. There is juvenility in citrus. You don't get any flowers till the plant becomes 4 or 5 years old often. And in California, we have a special situation where we have difficulty confirming tolerance or resistance to the disease. Because citrus is a highly regulated crop, and in California, we have many regulations about movement of citrus. So it becomes very challenging, and I have to do all of my pathogen experiments in a contained research facility or a biological safety level 3 facility. So which is kind of limiting. However, there are many opportunities. That is, we know that we can incorporate resistance from citrus relatives, and with back crosses, we also know that we can develop new genotypes with resistance traits introgressed already. And the time factor, that is a major thing, but there are ways around it. So if you have resistant hybrids, we can have a citrus industry which is easy to manage, and it's economically easy to develop, and you don't have to use too many pesticides, so it's ecologically acceptable, and it is a sustainable method of cultivation because you can control psyllids for only so long, and then after that you have to give up in spite of using so many pesticides. And then once you develop a variety through breeding, public acceptance is not an issue. People have no objection because it's not genetically modified in any way. So especially here, anything new has to be approved by the Food and Agriculture Department, Environmental Protection Agency, and those approvals are easier with our new genotypes. In Riverside, California, we have a citrus variety collection, which is like one of the largest in the world. We have about 1,200 varieties of citrus. This collection started about 120 years ago. At that time, it was possible to bring germplasm from different parts of the world. So we have varieties that you grow in Nellore, in Tirupati, in Mysore, Coorg, wherever. All of those also we have. Not everything, but we have the major ones. So this is a very valuable resource for us. What I wanted to point out to this audience is that the germplasm is available to other scientists and citrus industries all over the world. If you have valid permits and you make a request, you can get those. So I think we should all make use of what's available. Now the Australian limes that I'm using in the breeding program are shown here. One is a desert lime or Eremocitrus glauca. Then we use microcitrus species, Microcitrus australis, australasica, and there is another Inodora. Recently, the taxonomists want to call microcitrus as citrus. So when we publish as Citrus glauca sequence, Citrus australasica instead of microcitrus. But in my talk, I'm going to refer to it as microcitrus, just to distinguish this from citrus. So we generated hybrids of the first generation, or the F1 generation, first filial generation. And we observed that the F1 hybrids with the microcitrus parentage, they can stay in a disease ravaged place like Florida for many years. I have trees, F1 hybrids, in the field for over 12 years now, and they have no disease. So this gave us confidence that with this approach, we can get resistance that is sustainable in the field where the disease is rampant. So this is our breeding scheme. In the first round, we cross a mandarin with a microcitrus, and then the F1 hybrids, sometimes they have resistance, sometimes they don't have resistance, but either way, they're usually having off flavors. They're not really good to eat. But if you cross it back with citrus again, you can get some hybrids with citrus-like fruit and almost citrus-like flavor. Now, so this involves a lot of work, and to make the process go faster, we want to develop some genetic and genomic tools, because that will hasten the breeding process. This slide shows evaluation of plants for HLB resistance. We do these experiments in the greenhouse and also eventually in the field. In the greenhouse, we do challenge by grafting disease material. We also cover these individual plants with a sleeve, a mesh sleeve, so that we can introduce some psyllids inside. And this is called no choice feeding, because the psyllid has to feed on that specific plant or it has to die. And we know that the psyllid already has pathogen. So the pathogen goes into the plant and then you have a proper exposure. So we do have some susceptible ones, as you can see here in the top panel, or some resistant ones. We score hybrids for disease response on a scale of 1 to 10, depending on the bacterial titer in the sample, the time it takes to develop the symptoms, maintaining the bacterial titer, because sometimes the titer goes away, and also the field response to pathogen exposure. Now here I show 3 plants, 3 hybrids. This plant has a score of 2, a score of 4 and 10. A score of 1 indicates high level of susceptibility, whereas 10 represents resistance. So in the greenhouse, the evaluated plants only get a score of 1 to 8. And once they survive the field abuse, then we give them a score of 9 or 10. Now we extract DNA from these. Usually the DNA extraction is done in a high throughput manner. You can do 96 samples at a time in a couple of hours. And then we use a real-time PCR instrument to quantitative real-time PCR, so you can see what the bacterial titer is. So these are all routinely done. And it's important to define resistance and tolerance. We define resistance as a situation where the pathogen can be in the plant in a temporary way, but then the plant gets cured of it. So something in the plant kicks in, and then the pathogen is eliminated. So that's resistance. Whereas in tolerant plants, the pathogen does establish in the plant, although it doesn't go to very high titers. So you will get some yield from the grower's perspective, but it will not be very profitable. Now, HLB is a very slow disease. The pathogen is not uniformly distributed. So you need to do evaluations for an extended period of time, which itself is a challenge. And in California, as I mentioned before, we have additional challenges because of the regulations that we have. We are aiming to get something that's broad spectrum and durable, so that the hybrids that we generate will be useful. And it is very important that whatever we develop has tolerance in the field situation. So to develop the genomic tools, first of all, we sequence the genomes of the Australian limes. We sequence the same plants that we used for our breeding. So we have now sequenced several species of microcitrus and aromocitrus. Now these diverge from citrus by about 3 million years. Okay. So, and it is likely that the resistance genes that are there in these plants are not there in citrus. It's possible. So sequencing provides a lot of information about the genetic makeup. And now we have published sequences of Aromocitrus glauca, Microcitrus australasica, and Microcitrus inodora. Many more are in the pipeline. And these are the genomes are annotated. Then we also try to identify the resistance genes, which I'm going to talk about soon. So if you have molecular information, then it may be possible to identify potentially resistant plants while they are still in the greenhouse. So we don't have to grow them all out in the field to figure out. So at least we can do some kind of pre-selection. So once there is a specific candidate gene that is responsible, it will be possible to do plant transformations, perhaps targeted gene editing, and you can introduce resistance to elite cultivars. So the genome sequences that we generate are usually phase genomes, that is the two haplotypes sequences are separate. And then when we assemble the genome, it is de novo. That is, it's not based on, it's not matched to something that is already there. We just use that and then assemble it by itself. So, because if you compare it with something that is already there, a citrus that is already sequenced, and this has some novel fragments, you may miss those because they will not align. So these are chromosome scale assemblies. And Citrus australasica, glauca, and inodora are already published. The article came out in MDPI plants. And we have other Citrus australis and some of the susceptible mandarins that we have used. This paper is about to be published now. And we also sequence several other citrus parents that we have used and some of the hybrids that are considered essential. I just wanted to draw your attention to number 9 here. This is a pomelo from India, Caryoppa pomelo. In our collection, we also have this pomelo plant, and we are exposed to the situation in Coorg in Karnataka, where there was a thriving citrus industry once upon a time. But then because of disease, this industry went away. But there were some pomelo trees that did not get the disease. So one of them is available for us, and we are also using that. The reason for including this is because you want your resistance to be durable and broad spectrum. It should be resistant to several strains of the pathogen. And bringing in resistance from different sources is very useful for this. So for the genome assembly, we do genome sequencing, and then we do PacBio sequencing. Then we do scaffolding with another process called Hi-C, and then we do chromosome scale assemblies. Now, these genome assemblies can be compared with others. For example, here the Microcitrus australasica genome is compared with the clementine mandarin, with the trifoliate, and also with lemon. And the colors here represent 9 chromosomes that are there in citrus. And the cross lines here show that a fragment from chromosome 1 here has been moved to chromosome 8. Okay. So these are all important to do our analysis. With help of another collaborator who put in a lot of work on this, we have identified the R genes, or the resistance genes, that are known to be present and that are likely to be involved in the resistance process. So the resistance genes that we see in the Australian limes are different from those that you see in citrus. And this was also published in Frontiers in Plant Science recently. So the genome sequencing of 3 Australian limes is completed, and the genome sizes are indicated here. We have pretty good sequences, pretty good assemblies with very decent numbers. And you can also see that the R genes are shown here. This is in Australasica, and we have similar ones in Microcitrus australis, inodora, and so on. I wanted to show you some of the fruits that we get in our project. Now, the finger lime parent that is shown here is actually the parent. It's called finger lime because it's like a finger. It is a citrus, but it is like a finger. And when we use that as parent and cross it with citrus, you have a wide variety of phenotypes. We have many. I've just shown some representative ones here. See, this is bright orange. Everything is crossed with this and a mandarin. Okay? But you do get a lot of phenotypes, and it's very interesting to watch. And now we have hybrids of the next generation. Now, like I said, in the first generation, you do have a lot of off flavor that comes through. So we want to make sure that we retain the resistance trait, but improve the flavor so that it is more like citrus. Now, these are many hybrids of the second generation that we have fruiting now. And many of them are like citrus. I have hybrids that have fruit like pomelo and so on. These are also advanced hybrids of citrus with Australian lime parentage generated so far. Actually, this slide is a couple of years old. Every year we get newer hybrids, and it is very exciting when the fruiting season comes, because every year we don't know what we get, and we get different types of hybrids. So when we do a lot of characterization of these hybrids, so basically we take pictures of those, measure the juice percentage, see the Brix content, or how much sugar is there and how much acidity is there, and so on. We have hybrids that have mandarin-like flavor, hybrids that are lemon-like, grapefruit-like, and microcitrus-like. So the HLB tolerance studies are ongoing in greenhouses. We also do disease resistance studies in Florida and Texas, because what you study in the greenhouse has several limitations. So we also send to our collaborators in Florida and Texas, where they are able to plant it in the field and then observe how they behave when there is a lot of pathogen. In Florida, the field where we are growing these experimental plants have a lot of psyllid, and the psyllid is not controlled there because those are experimental fields. So if it can survive there, that means that's a good resistant plant. These are some other advanced hybrids that we have developed, and these are coming out now. And many of these are very high yielding. If they are acceptable in terms of resistance and fruit quality, then one of them will be a winner. These are selections, second generation hybrids with microcitrus parentage. So evaluation of several of these for horticultural traits and also for resistance is in progress in different states with my collaborators. We also conduct metabolomic analysis of the fruit juice to determine the organoleptic properties. That is, does it smell good? Does it smell right? Does it taste right? And so on. And we also want to identify compounds that impart characteristic flavors. For example, limes have several characteristic compounds that make them taste like a lime. So we want to know what type of hybrids we are generating. So that is another field of study. Now, as you can see, we have generated a lot of breeding data, and it is very difficult to manage with just an Excel sheet, which is what we were doing till recently. Till 2 years ago, I could remember all the hybrids that we have generated and what the parents are and so on. But now I have more than 1,000 trees in the field, and it's not easy to know what the traits are. So we use this breeding information management system that is developed by Dr. Suk Jung and Dory Main of Washington State University. Now, the thing is, these are freely available tools that anybody can use. And basically, you have like a field book app. It's like a small cell phone. It's like my cell phone, a tiny thing. And you can take it to the field and enter it there, enter the traits on your phone unit, Android phone unit, and then it can be transferred to another program on the computer. So it's very, very valuable, and we are still learning this, but this is extremely useful. We also get a lot of support from our collaborators, Suk and Ron Tapia. As we speak, Ron Tapia is coming here tomorrow to teach our new technician how to use this this Fieldbook app so that we can use it to the maximum. And this is also a free tool anybody in the world can use. So if you are doing breeding, I recommend this. Now, analysis of advanced hybrids. Once we have hybrids, what do we do? First thing I do is are they resistant or tolerant? So we do the testing in a greenhouse in California in our limited situation. And the second important thing is, does the hybrid have acceptable fruit? So we need to do taste evaluations. And then we need to determine the disease response in multiple field environments. As I said, we do it in California for horticultural traits. We also do it in Florida, and we also do it in Texas. Once we decide that some hybrid is valuable, then it will go through further scrutiny, and then we evaluate more. And in Florida, we have I have about. Did someone ask me a question or something? I'm sorry.
No, madam, please continue.
Yeah. So we do the PacBio sequencing and skim sequencing of a population of specific population of hybrids. That is, you determine a low depth sequence about 1.5 or 2.5x, and then use that to identify genomic fragments that may be correlated with resistance. Another aspect is the metabolomic analysis of the fruit juice, because we can associate off flavors with presence of certain compounds. So we will do the selection of hybrids that we know lack these compounds that are imparting the off flavor. So selection of breeding populations is based on presence of certain genome fragments, taste evaluations, other types of testing to see if they're likely to be resistant, and presence of resistance genes. So genetic variation between the individuals is important. Now, the most common variation in citrus is called a SNP, or a single nucleotide polymorphism change. Here, what this means is single nucleotide changes that are characteristic are often present in citrus. So although they are similar to each other, about 95 to 98%, there are small variations that determine if it's like a pomelo or a mandarin or a grapefruit or a lime. For example, here in microcitrus, in this position 13, you have a T, whereas in others you have a C. Similarly, in all mandarins, you have a T here, whereas you have a G here, and so on. So these are single nucleotide polymorphisms. So this is something that we study a lot because it's easy to study compared to several other variations like structural variations, which are much more complicated. So the citrus genome is about 300 million bases, and we know the genome sequence of many species of citrus. They're all available. I will show you a little more about that. And in our lab, my boss, Michael Roos, developed a citrus SNP chip with about 1.5 million markers. That is, it has 1.5 million small fragments of DNA on this gene chip. So if you have your particular hybrid, you can make DNA of that and then throw it on the chip and see where it hybridizes. That will give you a lot of information. Now, in addition to this 1.5 million marker chip, we also have a smaller version which has 56,000 markers. So these chips are used to distinguish different types of citrus and also to identify hybrids. And we can also document differences between individuals in a breeding population. If you can correlate with specific phenotypes or specific ways by which the citrus types differ, you can understand the genetic basis for that difference. Now, this is a process called Cas genotyping. So here, what we do is when we generate hybrids, we want to make sure that the hybrids are what we think they are, right? So when we do the crosses, we do remove emasculate the flower, that is remove the pollen, remove the stamens, and then put the pollen of our choice on the stigma. But you have to make sure that it is what you think it is, because in genetics, the mother is a certainty and the father is a probability. So we are doing this kind of genotyping for all our hybrids every year. First of all, it confirms the hybrid parentage because we know what the signature sequences of the parents are, so we can do that. And we can also identify if regions of interest are present. That is, if certain R genes or other loci that are presumed to be associated with traits that are significant are present or not. We can also map recombinants, and we can correlate recombinants and disease resistance if you have enough information. Now the thing here, the rectangular piece here, it's a 96 well plate with all these markers. And here what we do is we take the hybrid plants, just they take some discs, 6 discs of the hybrid plant, put it in each well, okay? And then we dry it. And actually I send this to a company to do the genotyping. We can do it in-house, but then it is much cheaper for us to outsource it to the company. Now, in this position, for example, if it has TT, it will map here. If it has CC, it will map here. If it's a hybrid with T and C, it will map here. So you can get this kind of information for each SNP, each marker. And when you combine a lot of these together, that will be a very useful information. Now the markers are identified for CASP genotyping of hybrid accessions. And this cartoon here shows the 9 chromosomes of citrus, and the lines represent the locations where we have designed the markers, the SNP markers, okay? And the red lines are for R gene markers, the blue lines are for some other types of genes that are known to be associated with resistance and so on. So we design the SNP assays. Every year we change it because depending on what is known, if new publication comes out saying something is important, then we try to incorporate that into our genotyping platform. So the genotyping patterns obtained are very useful to associate regions that are specific for the genome-assisted traits. So this slide shows genotyping of advanced hybrids. And here I have advanced hybrids, BC, Bacross, the second generation hybrids, hybrid 1 to 12. So these are 12 plants from one fruit, okay? 12 seeds from the same fruit. Now you can see how they are all different. So each box, each color represents one chromosome. So chromosomes 1, 2, 3 to 9. And all the colored regions indicate that that part of the genome came from microcitrus. The ones that are white show that it came from citrus. Of course, this doesn't have too much depth. It's only 86 markers in this particular cartoon, but it gives you an idea as to whether chromosome 3 is like citrus or is it like microcitrus. So this gives a lot of information. And here, the position, these are all the positions in the chip, citrus chip. I'm not using the citrus chip here. I'm just using the information developed in the citrus chip to develop these simple assays. It is not too expensive to do this, and a lot of information is already present. These are freely available, so anybody can use. We also use another resource called the Citrus Genome Database, which is again another free resource. This is managed by Dory Main's group. Dory Main is a scientist in Washington State University, and her team, they maintain this. Now, when we develop some new genome sequences, we deposit that in GenBank, in CBI GenBank, but they also go to this Citrus Genome Database, which is a much more user-friendly platform so that you can look at specific regions. We have also loaded our hybrid sequences here, whatever you see here. They were all loaded into this for our sake by our collaborators. We collaborate with this group. And we also get a lot of support from them. So if you are interested in looking at a particular gene, you can see how it is in the two parents. This is one parent, Australasica. This is another parent, Wilking Mandarin, and this is one of our hybrids. So how it is in the different ones, different hybrids and the parents, you can see here, and that is very useful. Now, often genetics is complicated. It's not simple. For example, here in this, I crossed a trifoliate, which is number 2 here, with a microcitrus, which is number 3. And this is the flower of trifoliate. This is the flower of microcitrus. But the hybrid I got has a fruit that's at least 5 to 6 times larger than the parents. Okay, and the flower is like that. That's because it's a multigenic trait. It's not one gene controlling this, but a combination of several things. So similarly, resistance is also determined by several genes. That's why it is very difficult, very challenging to do gene editing or anything, because first you need to know which gene to modify and how many genes are there. So that poses a lot of difficulty. Now, there are other important factors to understand about variation. So with genome sequencing, we can also obtain transcriptome analysis. We can look at the genome. We can look at the transcriptome, which is the RNA sequence. And then we can integrate the eQTL data. So we know that multiple SNPs may contribute to small but cumulative effects of the phenotype. So there may be several things that are required for you to get resistance that is meaningful. And often major agronomically beneficial traits are known to have multiple QTLs. Now, many of these variations like SNPs, insertions, deletions, or translocations can impact protein expression. And also the environmental changes can affect phenotypes. So it is a fairly complicated topic to study. We have another set of collaborators from Texas A&M University. They are doing RNA-seq of the hybrids, of the tolerant hybrids and susceptible hybrids. And this work is in progress again. When we have hybrid fruits, we want to see how they are. Are they like mandarins or pomelos or lemons? And if they have bad flavor, why is there bad flavor? So I have a postdoc who works entirely on that, Pallavi. So what we do is we extract juice from the hybrids, and then we are lucky to have a metabolomic score in our building. So the metabolomic score analyzes this. They pass it through HPLC, GC, whatever is needed. And then they generate a lot of data. Some of the data is targeted. That means you know what each peak corresponds to. And often it is untargeted. The untargeted seems to be very important for us because no one knows exactly what type of compounds are there in these Australian limes. They have not been studied that much. So there are many, many uses for this metabolomic study. One of them is we can identify certain compounds. If those compounds are present in high amounts, then it seems to have more of a microcitrus flavor. So we can get that kind of information. I wanted to point out something else here. See, in my crosses, I use this Wilking mandarin that has this purple oval here and crossed it with Microcitrus australasica. The F1 hybrid or the first generation hybrid is here, hybrid 71, okay, F1 hybrid. In the next round, I crossed the F1 hybrid with another mandarin, Algerian clementine, and then we got a population that is shown here, okay. One of the hybrids that has a fruit that is just like mandarin is clustered along with the mandarin here, okay. So such information will also help us direct our breeding efforts, because you don't know what to breed, which parents to choose and all that. So this is very informative. But again, metabolomics generates tons of data. So you need to be able to filter through and figure out what is it that is relevant to our study. Now, when we have hybrid fruits, we taste in the lab. Initially, we taste in the lab. Whoever is there will taste and say this is good or bad, stuff like that. But we also invite other citrus growers, citrus industry people. This is a grower in California, very much interested in our project. So he stops by every year, and then he will eat the fruit and give us his frank opinion, where it is and all that. So we also have other ways of monitoring the acceptability, what they call the hedonic score. And this I do with another flavor expert, Dr. Arpaya, who generally works on other fruits, but also helps us with our citrus tasting. And just in 3 days, we have a UCR Citrus Day, and we plan to give for tasting 3 fruits that we have selected this year to about 70 people. We label them as ABC, and then we ask them to score them to see how acceptable the new fruits are. So we have a lot of collaborators, and I think this is very important. So I can see the power of collaboration here. This is us in the center. We have the Texas A&M group. We are able to do field trials in Texas. We are ready to put about 1,000 trees in the field in Texas to see how they perform horticulturally, and also to see if they show tolerance or resistance. They also do other molecular work for us as needed. And the Washington State University people, Dory Main and her associates, they are the ones who also developed the breeding information systems. They maintain the citrus genome database, and they help us put our sequences there, and with a lot of technical expertise. Because the project has so many different aspects, there is no single person who knows all of them. But because we are able to collaborate with multiple people, we are able to make progress. And then we have this USDA Appalachian Fruit Institute in West Virginia. And so Dardick and his group, they're the ones who did the origin study. And they basically work with stone fruits, which are apples, plums, prune, etc. But the Rosaceae people are way more advanced than Rutaceae people. So we take advantage of their knowledge. And University of Florida and US Horticultural Lab Fort Pierce are very important collaborators, because that's where we have all our field trees, not all our field trees, part of our field trees that those field experiments tell us if the hybrids are really resistant and useful or not. We also do many other studies with these collaborators. And USDA Citrus Repository in Riverside is a major collaborator. They are very helpful in doing many things. They are part of all the things that we do here. And University of Tennessee, my collaborator there, Meg Stratton, is also doing many other aspects like a pangenome. That is, you compare genomes of several different types and then come up with something that is considered to be essential in all resistant plants and so on. So I'm not showing any of that data here. But we all have Zoom meetings periodically. Every month, so we exchange notes, and also every year I conduct a project meeting where everyone comes, including the industry people and our advisory board members, so that we make sure that we are on track and see how we can improve. So it's been working very well. This is to summarize our program in a nutshell. So we have like 10 major objectives. One of them is to identify sources of HLB resistance. I thought it was complete, but now we have introduced the Cariapa Pomelo. So that is an ongoing thing. And we think that having many sources of resistance and incorporating the essential traits into the hybrids will be extremely beneficial. Breeding novel citrus varieties. This we do every year during springtime. We do at least 1,500 crosses, and we continue the breeding. Genotyping is another continuous process, and every year the genotyping targets may be different. Sometimes it's the same, sometimes we incorporate new markers as needed. The pathogen challenge is the most important, and it is like a bottleneck for me, because sometimes it's difficult to see whether the plant is resistant or susceptible, and it takes several years to confirm resistance. Although we can confirm susceptibility early, to make sure that something is resistant and useful in the field situation is time-taking process. Now, genome sequencing of resistant varieties and also susceptible varieties, we have made considerable progress in this. And identification of important genes, we don't know. There are many important genes. It's not just 1 or 2 genes. Now, induction of early flowering and fruiting. A collaborator of mine tried this with the FT gene, but we failed miserably. We were not able to make the genotypes. And then the metabolomic analysis of fruit juice, as I described, that has also provided a lot of information that we are analyzing right now. The taste evaluations are also continuously ongoing, and then the field testing. So these are some of the new hybrids I got in 2024, but every year we get new hybrids, and then we have better selections all the time. I want to acknowledge my funders. Mostly the research work is funded by federal grants, USDA NIFA grants. We have been lucky to get multiple grants. Also, the local citrus research board in California funds the breeding projects. So they keep it going. And our project collaborators, there are many collaborators. The main ones are shown here. Ute Albrecht from Florida, Yang Ping Duan and Erin Raskoff from Florida, Chris Dardick from Virginia, West Virginia, Meg Staten from Tennessee, Dory Main and her group from Washington State University, Madhu Kunta and Park from Texas A&M, USDA Parlier, and Keremane Manjunath Keremane from USDA Riverside. And from UCR, my collaborators are Mike Cruz, Sean Cutler, Mary Lou Arpaya, Peggy Mark, and Khan. And the metabolomics core facility that is in UCR, they are our collaborators too, and they help us to understand what the metabolomics profile reveal and how we can interpret them. I have an excellent lab group: Kushwant, Pallavi, Lu In, Rayan, Koksal, Aisha, Manur, and many undergraduate students and high school students also collaborate with us. And so we hope that we will be able to get something that is useful to the industry sometime soon. Thank you so much.
Thank you, madam, for your excellent and nice presentation. And you covered all the advanced topics on citrus disease and greening disease resistance. And you already told about Australian lions are more tolerant to this Honglambing disease. And there is more 1,200 varieties of citrus. That is a great thing. That is more germplasm in your side. And also you covered many things regarding this breeding and genetics and genomics of this disease. And you covered complete genome sequencing, madam, and also management of breeding data and advanced hybrids and field book app. That is also new thing. And citrus genome is 300 million bases, and citrus SNP chip, it is also new thing for me. And also CASP genotyping and genome database, and the metabolomic analysis of juice from hybrids, and taste panels to determine the acceptability of hybrids. That is also a good thing, madam. And there is a new hybrids also developed in this citrus Honglambing disease resistance breeding. And thank you, madam, for your nice and excellent presentation. And now it is open for discussion. Any university officers or scientists or students may please be interacted with Dr. Chandrika, madam. Anyone from audience?
Madam, this is Dr. B. Govindarajulu from RHRS Tirupati. Very excellent presentation, madam, regarding Honglambing greening bacterium. This resistance breeding, what you are doing is highly appreciable. We are doing here regarding the standardization of protocols for citrus greening. And likewise, we developed this duplex PCR for both greening and mosaic also. And for standardization, we are using here the real-time PCR for greening. Likewise, the SOPs have been developed to detect citrus greening bacterium with I mean low filter of virus, or I mean bacteria. And for management, here we are doing 2 sprays of tetracycline hydrochloride 6 grams per 10 liters of water, twice at 1 month interval. And also we are giving a recommended dose of fertilizers along with zinc sulfate 200 grams per plant, that is the bearing plant, and ferrous sulfate 200 grams per plant for effective management of this greening. So these are the works we are doing here. Is there any, I mean, management practices regarding latest management practices regarding Hong Lang Bing green bacterium, madam?
When you said tetracycline, are you injecting tetracycline like they are doing in Florida?
No, madam. We are giving spraying and also soil drenching. No injections.
Oh, no injections. Okay.
Spraying, madam.
Okay, spraying. Here they have tried many different things, and for now, I think the tetracycline injection seems to be giving some result, but whether that is sustainable in the long term or not remains to be seen. They also have another way by which they can eliminate the disease. I wanted to include a picture of that, but I didn't do that. So they grow citrus under protective structures. So they have this huge structure which has a mesh. And the mesh the size is so small that the psyllid cannot go through. Psyllid cannot enter. So whatever is inside is protected. So apart from that, many others have been tried, many other methods, but nothing is really very useful. So we don't know. I mean, in some areas like in Florida, the disease came and it spread like wildfire. But in other places like in California and Texas, maybe it is a little slow. That could be because of the high temperatures or some other environmental character that we don't fully understand. So either way, because the disease is such a bad disease, we want to see if we can develop resistant plants that will be useful for California, Florida, and also for the rest of the world. So.
Okay, thank you, madam.
About your particular situation, maybe the nursery practices can be improved. You know, that is a different topic altogether, but that may be one option.
Yes, madam. We are also having the screen houses also, madam. We are trying to develop good planting material.
Yeah, that is crucial.
Thank you, madam.
Thank you, madam. Thank you, madam. Madam, Madam, Dr. Ramayam, sir, citrus registration, Petlur, madam, principal scientist and head, madam. Hello?
Yeah, go ahead.
Is it audible, madam?
Yeah, I can hear you.
Actually, worked up to 3 years in Tirupati citrus registration, madam. So at that time, the Hong Lang Bing, the virus, is it related to the temperature or humidity like that, madam? That is the environmental factors, because in high temperature condition, it is not detected in Tirupati area. So at that time, Dr. K. Gopal sir taken the samples to the Australia. There is a detection, madam. So my intention here is there any the pathogen or causal organism is related to the temperature, especially?
Yeah, the temperature may definitely have an effect. We know that because, as I said, in Texas and California, although the disease came here in 2012, it's now 2026. It has not spread as much as it did in Florida. And we believe it's because of, in addition to the other measures that we are taking, it could be because of the weather and the temperature. So
temperature play a major role. because in April, May condition, it is not detected here. But it is same 15 days gap, it is detected in Australia.
But when you don't detect
The
from
same campus.
plant, later on, when the weather is not that hot, are you able to detect from the tree? You said in May, when it is very
Yes,
hot,
madam.
you're not able to. You are
Okay.
able to detect, right?
Okay.
Are you able to detect the pathogen when the weather is cooler, is my question.
Yes, yes, madam.
Yes.
Okay.
So
So it is
when it is
purely
very hot,
dependent on the environmental factor also.
correct. When it is very hot, we think that perhaps it goes to the roots or something, and then it comes back when the weather is more congenial.
Okay.
So that
Okay.
is known.
Okay.
Yeah.
So because the vectors also active in summer prevailing conditions, because it is all sucking pests are more prevailing in on summer conditions, that is the high temperature conditions, but the more the disease transmission is also there.
Yeah, it is even in psyllids, even in the insects
Yes.
that are carrying the bacterium, when the temperatures are high,
Okay.
the psyllids will not have the pathogen.
Yes.
We have seen that because we had samples collected
Yes.
from actually from Pakistan. A graduate student brought some samples,
Okay.
and when they were collected in the peak
Okay.
summer, they had no pathogen.
Okay.
Okay, only when it is a cooler
Bacterium.
temperature they were coming in the plant. So the psyllid is not living that long, right? So it's fine, it goes away. But in the plant, you cannot detect in the canopy, but then it hibernates. It goes into the roots and stays there. So when the congenial conditions come
Okay.
back, then it goes to the canopy. So that is known. So just
Okay.
because you didn't see it for a while, it doesn't mean it is fully gone. So it is going to come back.
Okay. Okay. Thank you. Thank you very much, madam. Thank you very much, madam.
Madam, this is Dr. C. Madhumati. Madam, thank you for your very informative and insightful lecture. And it is truly excellent and easy to understand to all the students. So earlier in Citrus Research Station Tirupati, there are introductions from several exotic citrus sweet orange types like pineapple, Jaffa, Valencia late, Valencia like that. But right now the prevailing or leading variety is sweet orange South Goody variety, and a few varieties are from Jain and few from like Malta. Some of the varieties are also there in the field because of the private nurseries. So, madam, in your opinion, what are the suggestions for improving the diversity in the sweet orange varieties? What is your opinion to improve this genetic diversity in sweet orange? And one more question is, so Nucella embryoni, especially in acid lime, so we are recommending only seedlings to avoid this viruses and all. Because of the high polyembryonic nature in the acid lab. So is this citrus greening is also a seed transmitted issues are there with this? Or,
no, it's not known to be seed transmitted. Greening is not known to be seed transmitted.
So I need your suggestions to improve this citrus industry in Andhra Pradesh. Because this is citrus greening and dry root rot. So every year, as in Florida, we are losing the orchards with 5 to 10% drying of the trees.
I think the first step probably is in the nursery situation. There should be practices, and whatever is distributed to the growers should be free of diseases. You don't want to give plants that are already infected. Those should not be planted. No, it should be free of diseases, and there are methods of testing and all that, and those should be done. So I think sometimes I've seen greenhouses in India, and you have beautiful structures, but all the rules are not followed, and sometimes that can lead to lapses.
Actually, here so many private nurseries are doing budding and selling. There is no regulation. And farmers are also not, I cannot say aware, but they will depend on the local nurseries, which were not doing this virus indexed mother cyan blocks and all. That facilities are not. That is
Yeah,
also
so
another problem for
I believe there should be regulation. Rules are for convenience. Rules are for making things better. It's not just to make your life hard. So there should be rules and they should be followed. That will help.
Thank you, madam. Nice presentation. Informative.
Thanks,
Very
Anil.
informative. Thank you.
Any scientists? Students?
Madam, good morning, madam.
Good morning.
Myself, Dr. Bhanumurthy from Tirupati Citrus Research Station, ma'am. Ma'am, regarding your presentation, it is a nice and very elaborate, madam. Thank you for that. So, for using for any breeding cycle, madam, what may be the age of the parents, madam, to use in the breeding cycle?
Oh, age doesn't matter. To select the parents, one thing is your parent should be flowering, of course, otherwise you can't get pollen. And for the female parent, we use certain criteria, like I want a plant that doesn't have alternate bearing, which you have in some genotypes. And then we want a plant that is healthy and it is able to produce a lot. So you can, have part of them working for you the way you like and stuff like that. But with all that, with all the information we have, we have the breeding season coming up in another 1 month. So this 1 month, we struggle over it, ponder and gather all the information we can and make educated guesses, so-called educated guesses, but we are wrong 50% of the time. So we just do more crosses than what we need, so that even if you have some failures, you will hopefully have some successes in the direction that you want. Because often when you read the literature, they say they have a common, they describe a common trait for all pomelos. You cannot have all pomelos in one group like that. There'll be lots of differences. And no one has studied all the pomelos, right? So there is that element of surprise, and we are prepared for it. That's it. It just means a little more work, and so be it. That's not a bad thing.
Thank you, madam. Thank you.
Hello, ma'am. This is Tanuja, ma'am.
Hi, Tanuja.
How are you, ma'am?
Good. Nice to talk to you.
Yeah, ma'am. Ma'am, you were mentioning about that finger lime, where you are taking it as a parent.
Correct.
And another thing, like Australian limes, like where we are using, usually we are doing work on the sweet orange. But in case of mandarin, where you are focusing more on the mandarins. And recently, Dr. Ghosh sir has brought so many gem blossom from your place. And is there any sweet orange collections he was included, or
any sweet oranges in what Ghosh has taken?
Yes. We don't know, ma'am. Just I'm asking.
Yeah, I'm not aware of the what exact genotypes he has taken, but we do have several sweet oranges here.
They both Jain group also has.
Yeah, even the Jain group in Jalgaon, they also have taken some.
Okay.
So there is this citrus variety collection site. So if you go to that,
okay, ma'am.
Citrus variety collection Riverside. Okay, so if you go to that, there is a list of all the genotypes that are there, and also a little about each genotype, where it came from, what type of fruit it gives, in which season you get the fruit, and what is the acidity, what is the sugar content, and stuff like that. There's a lot of information out there. So this is the variety collection side that I showed you.
Okay.
But whether it will perform well in Tirupati or not, that we don't know. I mean, that has to be tested, because maybe certain varieties perform well here, and they may not perform equally well in Tirupati. That is possible. So.
Okay. And what is that speciality, characteristic nature of that finger lime, what you're mentioning?
Oh, I showed you a picture. I think it's like a finger. It's not microcitrus, right? It is actually, they do eat it in Australia. They do make different types of foods with it, and they extract the vesicles and add it in specialty restaurants to, I guess, some fish dishes or whatever. So it is edible, but it doesn't have the same flavor as citrus. So our interest in microcitrus is only to get the resistance trait.
Okay. Thank you, ma'am. Thank you.
Madam, I have one basic doubt. How you are maintaining the germplasms in Florida, madam? Any because the open field will have these citrus greening issues. So any precautions or any measures are taken in maintaining the germplasm?
No, germplasm is not maintained there. Germplasm maintenance is in California. California, okay? In Florida, what I'm doing is, I want them to be infected by HLB. I want to see which is the survivor. Okay, so there we are not maintaining germplasm, only in California. California, we do have the disease, but it is in isolated pockets only. And our variety collection so far, touch wood, so far it's been free from HLB. In some areas near Los Angeles, there are areas with the disease, but the California Department of Food and Agriculture is very, very proactive, and they keep monitoring the situation. And if they see one diseased tree, HLB diseased tree, they pull it out, they burn it, and they monitor the trees, 800 feet around that and all that for several years. Spray, make sure there are no psyllids. They do a lot of control, and they spend a lot of money. Numerous dollars go into that. But that has paid off, because the first disease report was in 2012. Now we are in 2026, and it has not spread much. I think that is because they have done a good job. But still, it may not be adequate in the long run. If it spreads to the Central Valley, where there is most commercial citrus, that may not be adequate. And all this we were able to do only because Florida had the disease, and Florida shared information with us. And we learned a lot because of our colleagues in Florida. And the California industry adopted those and followed those. That's why. See, everyone doesn't have to, find everything. You can also learn from other people, So California did that. So that was good.
Thank you, madam.
Madam, one more, madam, please, madam. So, Dr. Ramaya. Hello.
Chapandi.
Madam. So, one more thing, madam. Here, Dr. Sheshadri sir tried. Actually, the rootstock is Rangapur lime normally, but he has tried in wood apple, madam. It survives well, even though there is a symptom is there. It is survive well in wood apple. So then, what about the greening characters, madam? Is there any prolonged study on the change the rootstock, madam?
Rootstock, actually, they have done a lot of studies. One of my collaborators, Ute Albrecht from University of Florida, has done a lot on rootstocks. They do have rootstocks that are fully susceptible and some that are not so susceptible. But I don't think they have anything that is totally resistant. That is not known yet.
Okay, madam. Yeah. Because Sheshadri sir, he has very good interest on growing on wood apple, madam. It is successfully grafted. It is growing, madam. But there is no lengthy growth. But it is stunted growth. The growth is arrested not for a long time, madam. So
yeah.
That is
so wood apple is Feronia elephantanum.
Madam,
Velagapandu.
Velagapandu. Yes.
I know. Feronia elephantanum.
Yes. Yes, right, madam. But the plant growth is not so good, but it is stunted growth. But then what about the Honglang beans, madam? It is there is a spreading or not, or is there any resistance like that? But is there any information, madam?
Feronia, actually, I have seen one report, I think from Indonesia or Malaysia or somewhere. They have reported the pathogen, in that, but I don't think there are many studies. We do have Feronia in our collection, but I don't think anyone has studied. They don't even know the importance of Feronia. So.
Okay, madam. Okay, but the growth is very good, madam. Physically, I have observed there. I still now there is such grafting is there, madam. But it is a stunted growth.
Stunted growth, yeah.
Yes.
And do you think the stunted growth is because of HLB or some other reason maybe?
It is having a dwarf nature.
It is not stunted growth, madam. It is dark nature, and
stage.
the wood apple rootstock is used here, ma'am. Sir told it is sweet orange as the rootstock.
Oh.
But here rootstock is wood apple.
Oh, wood apple is the rootstock.
Yes,
I
yes,
see.
yes.
Wood apple is the root instead of
Wood apple, sweet orange was grafted.
Sweet orange good.
Madam, here we are using the Rangpur line rootstock and Alimo rootstock. It is also good for controlling this green, madam. And also we have Tirupati sour orange, and also we have Texas seedlings. We are trying for the resistance to citrus green. CRS, citrus research station.
Yeah. As far as I know, they don't have any resistant trifoliate rootstock or sour orange rootstock or anything like that so far. There are some some newer hybrids that have some tolerance, perhaps, but not really resistance.
Okay, madam. Okay, madam.
Madam, is there possibility to give this Australian lines, resistant lines to our university, madam?
You mean the germplasm, whatever I showed, and whatever is in the germplasm list. If you go to citrus variety collection, you can see the list of germplasm. Those you can import under permit. If you have the right permits, if you want, you can contact me and I can direct you to the right person. But the hybrids that I am developing, I cannot share with anybody because I don't own them. The university owns them. So even if they are resistant, I mean, we are still evaluating stuff. But even if I find something that is resistant, the University of California, they own it. I don't own it. So that becomes a little challenging. And that is down the line. So,
Ma'am, can we get the microcitron saplings or some source through proper channel?
Microcitrus is possible for you to get because that's part of the germplasm collection.
What about finger lime, ma'am?
Yeah, you can get finger lime also. There are finger lime varieties that you can get. And I put up my email address. I think Ruth madam has my email address. She can share with you. Please contact me and I will tell you who to contact to get the finger limes that are there in the germplasm collection, not the experimental ones that we are dealing with, because those we need to confirm, and also those I do not have the freedom to give them away.
Madam, what is this psyllid traps? Where it?
Psyllid traps. I don't have that project anymore, but we developed psyllid traps. So the psyllids go there and get captured. So this was actually for people like growers to monitor the psyllids in their grow. So we developed these psyllid traps. They could hang them in their grows, collect the psyllids, and also do a testing. We also developed a testing system using a loop mediated amplification method. So it's like a small unit that they can use and do the testing just to see if there is HLB or not. Because if you know early on that you have the pathogen or the disease, then maybe there can be some mitigation strategies, disease mitigation strategies that can be followed. That was the idea. That was a project. I don't have that project anymore.
Thank you. Anybody? Students?
One more thing.
Sir, just Naik, please. Myself, Dr. Ramanandam, Director of Research, Dr. YSR Horticulture University. Especially on citrus, the pioneering institute is our citrus research station, Tirupati, where they have developed excellent varieties and technologies for improving the production and productivity, and also disease management. Madam, actually, our scientists asked that exchange of varieties or importing of varieties from your institute and everything. Our university has almost had more than almost 100 MOUs with within the country and abroad. So, is there any possibility to have an MOU with your university, especially for sharing of ideas or some collaborative research in citrus?
Well, for germplasm collection, I told you if you write to me what you want, and I cannot send it, but I can direct you to the organization that sends it to you. Okay, about other MOUs, you have to contact the university, with the university people.
I request the Director, Industrial and International Programs, Dr. Ruth, you just try to pursue this matter, and you can have the MOU. If you can have the MOU with that university, then everything in that MOU, we can have the exchange of varieties or some, this collaborative research, etc. in our university also.
Yes, sir. Yes, sir.
You can discuss, we can discuss with our Honorable Vice Chancellor, and we can have one program as Madam suggested. Then we can approach the university authorities so that it will be useful.
Yes, sir. We
for
will
us.
pursue, sir. We will pursue.
Thank you, Madam, for your excellent presentation and very informative. And I think all our scientists are enriched regarding this citrus greening disease and the progress of work made at your university.
Thank you.
So definitely it will be very much useful to our scientific fraternity. Thank you very much, Madam.
Thank you.
Anybody from audience? Anyone?
Kindly contact other scientists if you have any queries. Madam is ready to spare her valuable time to clarify the doubts.
Sir, nobody is there. I will invite our registrar madam for concluding remarks. Please, madam, Madhavi madam.
Madam, I think you can request our Honorable VC sir for his remarks after the presentation. If not, I'll proceed.
Sir.
Okay. Thank you, madam. Excellent presentation. And many things, you have covered all the things that the molecular biology and what of the pest and especially greening is a major problem in Florida as well as in India. So definitely, as our Director of Research said, if any possibility is there, definitely we will have one MOU, and we will write a letter to your university, and from that side you can follow up, and because that exchange of any plant material, germplasm, that resistant varieties, and we can have we can share the technology, and we can share the germplasm. For that, we should have MOU, and if any possibility, kindly see that, and definitely our teachers, scientists will utilize your knowledge in future, and kindly share your WhatsApp number, phone number, and email ID. Any doubts they will get, they will share with you. And thank you very much, madam, really a wonderful lesson we have heard, and excellent technology. Whatever the work you have done, we enjoyed your teaching lesson. We feel that you're even though you're staying 20 years in USA, we feel that madam is our madam, and from Mangalagiri, that I think native place Mangalagiri.
Yeah.
You are in sari and all, excellent, very nice. And we are not treating you as a foreigner. So you are Adapaduchu. So we feel like that, and you
Daughter
will
of the
have more
soil.
response.
Daughter of the soil, I said.
Yeah. So you have to help our university also from your side, that collaborative research projects, training programs, your lab facilities, library facilities for our teachers and students. So definitely we'll have many things in future. So first of all, we plan for MOU. Thank you for giving wonderful lecture, and again thank you from Horticulture University, and we'll have future relations maintained between our two universities, Dr. YSR Horticulture University and University of California. Thank you.
Thank you.
Over to Madam Registrar Garu.
Thank you, sir. Respected Chairperson, Dr. K. Dhananjay Rao, Honorable Vice Chancellor, esteemed university officers, distinguished speaker, Dr. Chandrika Ramadugu, project scientist, Department of Botany and Plant Sciences, University of California, Riverside, California, USA, associate deans, associate directors of research, scientists, faculty members, researchers, and my dear students, very good afternoon. On behalf of organizing committee, I have the privilege of proposing the vote of thanks for this insightful seminar on citrus Honglongbing use of breeding, genetics and genomics to develop novel varieties with disease resistance. First and foremost, I express my sincere gratitude to Honorable Vice Chancellor for his gracious presence, guidance and support rendered in organizing the seminar. I express my sincere thanks to esteemed speaker, Dr. Chandrika Ramadugu, for the enlightening presentation on the challenges posed by citrus Honglongbing, the advanced studies being carried out in breeding, genetics, and genomics to combat this devastating disease. Madam, your expertise has enriched the understanding of developed advanced hybrids of citrus using different citrus species and closely related species as parentage, along with various steps involved in research program. HLB continues to threaten citrus production in countries like India, Brazil, United States, and many other citrus growing countries, making today's deliberation highly relevant and timely. I extend my heartfelt thanks to the university officers for valuable insights and thoughtful observations. My sincere appreciations goes to all scientists, faculty, students for their involvement, discussions, and interactions. I also acknowledge the dedicated efforts of the organizing team, which ensured the smooth conduct of the seminar. I hope that the knowledge shared today will strengthen our efforts to work on HLB resistant citrus varieties through advanced breeding and genomic interventions. With gratitude to everyone present here, I conclude the vote of thanks. Thank you, one and all.
On behalf of Dr. YSRHU, big thanks to our guest speaker, Dr. Chandrika Madam, and we are thankful to our Honorable Vice Chancellor Sir and the Registrar Madam and all university officers and the faculty members of Dr. YSRHU and all the students of Dr. YSRHU and the supporting staff from DIC section. And thank you once again. Now the program is closed with the permission of Chairman. Thank you, sir. Thank you, one and all.
Thank you, madam.
Thank you.